Hello, everyone. I'm Vicki McCullough, the Southeastern Account Manager at Cell Signaling Technology, and welcome to today's CST Learning Lab live session. I'd like to introduce Dr. Fang Chen, who is one of our cut-and-run scientists at Cell Signaling Technology. Dr. Chen helps develop and validate reagents and antibodies for use in epigenetic studies, especially for protein DNA interaction studies. Today, we are going to discuss reasons to try cut-and-run to analyze protein DNA interactions. During today's session, you will also have the opportunity to ask questions by typing them into the GoToWebinar questions area in your control panel. For those of you who are just joining us, welcome to the CST Learning Lab live session. Without further ado, I'll turn it over to our cut-and-run expert, Dr. Fang Chen.
Hi, good morning. I hope everyone can see my slide deck very well. Give me a moment. Great. So I'm Fang Chen, the Senior Development Scientist at CST. I'm very glad to talk about CUT&RUN assay today. So as introduced by Vicki, today I would like to focus on what CUT&RUN is and what its benefits are. I would also like to show you some validation data for our CUT&RUN assay kit. As you know, just like CHIP, CUT&RUN is a powerful technique to review the protein-DNA interaction within chromatin. I hope to quickly go through the main steps of CUT&RUN so we can understand where the benefits of CUT&RUN come from. At the beginning of the experiment, we bind cells to the Concanavalin A beads to facilitate buffer change and cell wash. We have a very small amount of cells in CUT&RUN, so they are very easy to get lost during the spinning and the buffer change if not bound with beads. In CUT&RUN, the cells don't need to be fixed. We use digitonin to permeabilize the cells. The digitonin can only permeabilize cellular membrane but not the nuclear membrane. Therefore, in CUT&RUN, the nuclear membrane remains intact during the whole experiment. Both antibody and the protein A, protein G micrococoa nucleus enter the nucleus through the nuclear pores. Inside the nucleus, the antibody will bring the enzyme to the target protein binding site through the interaction between antibody and the protein A and protein G. Then we initiate the digestion by activating the enzyme using the QSM. The small protein DNA complex can diffuse into supernatant through the nuclear pores, while the large undigested chromatin will remain in the nucleus. Now you understand why we don't want the nuclear membrane to be permeabilized at the beginning, because we don't want the large chromatin fragments to leak out from the nucleus and produce high background signal. Now all we need to do is to purify the DNA in the supernatant, and assay is done. Clearly, CUT&RUN has no IP procedure. Without the low efficient IP step, we can significantly increase the sensitivity of the assay from millions of cells to 100,000 cells per action. By doing that, we can save a lot of time and resources on tissue culture work. Also, because CUT&RUN skips the chromatin prep and IP procedures, we can get CUT&RUN results much more quickly. It only needs 1 to 2 days from cell to DNA, while CHIP requires 2 to 3 days, so it saves our bench time. In CUT&RUN, the large chromatin DNA fragments are trapped inside the nucleus, so CUT&RUN doesn't need to sequence the DNA fragments from the whole genome. It only needs to sequence the digested small DNA fragments that are diffused into the supernatant. Therefore, only 3 to 5 million reads per sample will give a good signal-to-noise ratio with low background, compared to a typical ChIP-seq experiment, which needs 10 to 35 million reads per sample. That will significantly decrease the NGS cost. Due to the lack of cross-link step, CUT&RUN is working on native but not fixed chromatin. Then artifacts from cross-link will be avoided. Also, CUT&RUN is compatible with spiking DNA strategy, which allows for sample normalization and bring us more confidence on the result. We also have validated our CUT&RUN assay kit on different target types. transcript factors, cofactors, and histone modifications. So you don't need separate protocols or kits for different target types. Last but not least, our enzyme is compatible with both rabbit and mouse because it fused with both protein A and protein G. No secondary antibody is needed. Actually, there is one more point. The CUT&RUN reaction volume is much smaller than CHIP one. So typically, you only need 1 to 2 microliter antibody per reaction, while CHIP requires 5 to 10 microliter antibodies. That's another saving on the cost of your experiment. When starting protein-DNA interaction, no matter CHIP or CUT&RUN, different target types bring us different level of challenges on the experiment. The first category is histone modifications. There's very stable binding to DNA, and there's always a large number of histones per DNA fragment. So they are relatively easy to CHIP or CUT&RUN. The second class is DNA binding transcription factors. So they are less stable bind than histones, and there's always fewer factors bound per DNA fragment. So they are more difficult than histones. However, the most difficult type is the transcription cofactors. So they often don't directly contact DNA, but associate with DNA through protein-protein interactions. They are often involved in large protein complexes, so the antibody epitopes can be buried for antibodies. So it's very difficult to study its interaction with the DNA. People also feel worried for the cofactors in the CUT&RUN assay because they don't know if a large protein complex can diffuse through the nuclear pores by itself or not. So next, I would like to show you at least one example of each target type with our CUT&RUN assay kit. The first example is a histone modification antibody H3K4 trimethyl, the easiest target type. You can see that CUT&RUN assay works well with both QPCR assay and NGS assay. We include CHIP data here as well for your reference. So first, let's look at QPCR data. You can see that the QPCR signal from CUT&RUN assay and from CHIP assay are very similar. Same in the NGS result. The top track is CUT&RUN data, while the middle track is CHIP data. On both zoom out view of a whole chromosome and the zoom in view of no target genes, the binding pattern of peaks between two assays are same. But please keep in mind that CUT&RUN only starts with 100,000 cells, 40 folds less starting materials. The second sample is a DNA binding protein, CTCF. The function of CTCF doesn't like a typical transcript factor, but it directly binds to DNA just like all of the transcript factors do. Its binding with DNA is weaker than histones, but stronger than cofactors. Again, just like CHIP assay, the CUT&RUN assay can generate decent and reliable signals in both QPCR and NGS results. Next, let's look at cofactor, the most difficult target types. The example here, RPB1, is a component of RNA polymerase II, which size is 500 kD. The left QPCR panel is QPCR data, while the right QPCR panel is CHIP data. Clearly, CUT&RUN works very well for the cofactor RPB1. Same conclusion can be made based on NGS data. The top track is CUT&RUN-seq data. As cofactor is the most difficult target type and the researchers have most concern on this type of proteins, so I'd like to show you a couple of more examples of cofactors with our CUT&RUN kit. Cofactors SMART-CC1 and BRG1 are two components in the BAF complex, which is larger than 1000 kD. You'll see that they both exhibit binding peaks in the CUT&RUN assay, and their binding pattern match to each other, which is consistent with their biological function within the genome. Similarly, cofactors SUS12 and EZH2 are involved in the PRC2 complex, which is larger than 550 kD. Again, the binding pattern of two components in the same protein complex match each other. I'd like to mention that we actually tried a specific protocol for a target type of cofactor. We try to completely break open nuclear membrane, purify all digested and undigested DNA, and only purify the small DNA fragments by site selection. However, we always got high background using this protocol due to the breaking open of the nuclear membrane. Feedbacks from other labs also support our conclusion that this protocol doesn't work as well as it was expected. So I just want to show you the validation efforts behind our recommended protocol. I don't think it worth your time to further modify the protocol for the cofactors. As long as you pick a high performance antibody and perform the experiment carefully, you should be able to get good results for cofactors. Lastly, I'd like to show you some cell titration data. You can clearly see that even with 12,000 cells per action, we can still see a decent signal to noise ratio with our CUT&RUN assay kit. Okay, that's what I want to talk at the beginning of our today's Learning Lab event. If you need more information about CUT&RUN assay or CUT&RUN validated antibodies, please visit our websites. You can also reach out to us for technical support regarding to advice on selecting antibodies, assistance with your CUT&RUN assay assays, and the requirement for custom antibody formulation. Thank you very much. Now I'm open to your questions.
Thank you, Fang. For our first question, what should I do if I am performing a time course and can't immediately perform the CUT&RUN assay on all of my cell samples? Can I freeze down my cells? Does CUT&RUN work with formaldehyde fixed cells?
That's a very good question. We haven't tested our kit with fixed cells yet, but in theory, yes. As Hennicoff's paper indicated, CUT&RUN assay works with fixed cells. But actually, in this case, I suggest you collecting your samples in a different way. For example, if you need to treat your cells for 1 hour and 6 hours with a drug, I suggest you first start the 6-hour treatment, and then in 5 hours, you start the 1-hour treatment. So at the end of the 6 hours, you will get both samples ready for collecting. Yeah, next question.
If I am looking at the binding of indirect DNA binding proteins, such as transcription cofactors, do I fix my cells to stabilize the binding of these proteins?
So as I have shown you in the slide deck, so current recommended protocol is okay with cofactors, but if you really want to try the fixation for the cofactors, you can try, but you may need to do your own optimization for fixed cells.
I am working with a limited number of cells and looking at both histone modifications and transcription factor binding. What is the minimal number of cells I should use for my experiments?
Our kit is validated for 100,000 cells per action. And as I showed in the last slide of my slide deck, so the kit also works very well for about 10,000 cells per action. So although we cannot guarantee the kit works for a very low cell amount, I would like to give you a reference here. So we hope you can use at least 500 cells for histone antibodies, 1,000 cells for transcript factor antibodies, and 5,000 antibodies for Cofactor antibodies. I'd like to say the more cells you can use, the better. Don't challenge yourself with extremely low cell number if you do have access to more cells.
Do we need to run serum starved cells before performing CUT&RUN like we do before ChIP-seq?
No, I don't think so. So treatment is depending on like how your target protein bind to the DNA. If the binding is serum starved and induced, if not, I don't think it's necessary.
Does it also work well with tissues like brain? And do we need to optimize the manganese activation, or is it a fixed protocol?
As I said, we haven't tried our protocol with tissues yet. But I actually suggest you fix your tissue samples before the CUT&RUN assay, because the cell isolation from tissue is a relatively long process, and the molecular event inside the nucleus can change during this long process. So you may get a more accurate snapshot of protein DNA interaction if you fix your tissue before you do the CUT&RUN assay. And also optimize the PAG micronucleus activation. I don't think it's necessary. So I do want to mention that we don't suggest you to do any like optimization on the amount of the enzyme, and also do some time course on the digestion time, because we already tested different conditions and we know they are very flexible, and likely you don't get very different results by changing the protocol in these ways.
Do you have any advice regarding working with adherent cells versus cells in suspension?
They have no difference. So they are same. Like for adherent cells, you just need to like detach the cells first before you wash your cells. Suspension cells is even easier. You just collect it. That's it. So they don't show any difference in our hand.
Okay. Is it important that the antibody works in immunoprecipitation?
It's not. So that's kind of about antibody selection. So I want to talk more here. Like I want to say, based on our experience, like not all of the ChIP-seq antibody works in CUT&RUN, just like not all of ChIP antibody works in ChIP-seq, not all of the IP antibody works in ChIP. So of course, I first recommend you use a CUT&RUN validated antibody. CSTA already has some validated CUT&RUN antibody for you to choose. But you can, if you cannot find it, you can try ChIP-seq antibody. And if you cannot even find a ChIP-seq antibody, you can try IF antibody in the CUT&RUN assay, because both IF and the CUT&RUN works on living cells. So it's not necessary that the antibody should work in IP before it can work in CUT&RUN. And vice versa, even the antibody works in IP, it doesn't mean it must work in CUT&RUN. So it's not related.
How convinced are you that the antibody is directing eminase and you are not just mapping hypersensitivity sites?
That's a very good question. Nobody really knows this, because you know the CUT&RUN work on like native cells. So in theory, the molecular event inside nucleus can change during the experiment, right, before the digestion is performed. So I feel that the CUT&RUN is working on like more dynamic protein DNA interactions, while the ChIP is working on a snapshot, which may include more hypersensitivity sites. But yeah, so if you feel you want to use CUT&RUN and you want to catch everything, even the weak ones, you can try to fix your cells before the CUT&RUN.
See if we have any. Do you have any advice for the spike in DNA?
Spiking DNA can be used for both QPCR and NGS assays. And the amount of spiking DNA required in two assays are very different. So you'd better follow our protocol on the spiking DNA usage. And we have very detailed information on how much spiking DNA to use in each detection method, and how to normalize the signal in separate method.
Is cut-and-run analysis by QPCR as accurate as ChIP QPCR?
I think so, yeah. But you probably will see, sometimes you probably will see a little bit higher IgG background in cut-and-run assay than in ChIP assay, because sometimes the large DNA fragment also leak out from nucleus during the experiment. It's possible, but it's not too high, like slightly higher than ChIP. But that said, like you should always see strong signal-to-noise ratio of about 5-folds between your signal and IgG signal.
What negative controls do you recommend for doing cut and run followed by qPCR? What about in next generation sequencing?
So for QPCR, we recommend the IgG as a negative control. For the NGS, we recommend input as the negative control. Actually, both IgG and NGS are used in, both IgG and input samples can be used in NGS, but we just recommend input sample over IgG because it requires less reagent and it has more diversity. And actually, we already get some feedback from customers that IgG antibodies sometimes produce background in the NGS.
What advice do you have for qPCR primer design, especially if you can't meet recommended criteria? What parameters are flexible?
So of course, for the primer set, like first you need to follow the requirements for primers. For regular qPCR, no dimers, no hairpin structure, something like that. And you also need to pay attention to the size of the applicant, because the CUTRUN DNA is smaller than the chip DNA. So for transcript factor and cofactors, the typical size of the CUTRUN DNA is around 80 BP. And for histones, the CUTRUN DNA is around 150 BP. So if you can design your primer with an applicant size between 60 to 80 BP, you are sure that you can capture all the signals. But if you cannot do it, I think it's pretty flexible. Like I feel in our hands, like applicant size around 100 BP is okay too, especially if you are using it in a side-by-side comparison. So it's still a fair comparison.
What control would you do before sending your sample out for sequencing?
We usually do qPCR to make sure we see expected signal-to-noise ratio.
So the next question is, do you have any recommendations for the library prep kit?
I have a lot of recommendations for library prep kit, but the good thing is that they are all listed in our CUTRUN protocol, which is free to download on our website. Some main points is that you don't want to, when you ligate your adapter, you don't want to incubate your sample as very high degree, like 65 degrees. That will make your, because CUTRUN DNA is smaller, is shorter than chip DNA. So if you incubate a sample at like 65 degrees, so the double DNA strand will separate you like single strand DNA. So you cannot really ligate your adapter to your samples. And also to avoid the large fragments in your final library DNA, you don't want to, in the PCR amplification, you don't want to like extend the DNA for like too long, like say 75 seconds, maybe 10 to 50 seconds is enough.
Do you recommend performing a size selection step during library preparation?
No, we don't recommend. No matter for chip or CUTRUN, we never perform size selection. So in theory, size selection only get rid of the large fragments, but in fact, even the small DNA fragments can get lost. Our experience is that the large DNA fragments will not affect the sequencing on the small fragments in the same sample if you don't have a lot large DNA fragments in your sample. So it should be fine. As I said, if you really concern the large DNA fragments during the PCR amplification of the library DNA, shorten the extension time from 75 seconds to 10 to 15 seconds. That will get rid of the large DNA fragments.
Is cut-and-run analyzed by qPCR as accurate as chip qPCR?
I think I just answered that question. I think yes, yeah.
Is there a maximum number of cells that can be used in the cut-and-run assay?
That's a very good question. We tested 100,000 cells, but it should, based on the customer feedback, it should work on 200,000 cells. We didn't test like millions of cells in one reaction. But I don't think it's necessary, right? No.
Have you done cut and run on epitope tagged endogenous targets?
I don't, but some customers did, and I know they get good results. So you can try.
What about cut and tag? Can you recommend one of the methods over the other, cut and run or cut and tag, in general or in a particular setting?
Yeah, so unlike the CUT&RUN, CUT&TAG use the TN5 in the assays. So, you know, in CUT&RUN, the DNA is digested, but in CUT&TAG, TN5 cannot only digest the DNA, but also ligate barcode DNA to the DNA fragments. So it saves time for library construction. But CUT&TAG is not as sensitive as CUT&RUN, so it needs to use the secondary antibody to boost the signal. CUT&TAG works for single cells, so if you want to do single cell, maybe you can try CUT&TAG. But otherwise, I think CUT&RUN is sensitive enough, and it should have higher success rate.
How do you recommend testing and optimizing an unvalidated antibody in your cut and run assay?
Well, we suggest testing it alongside with the positive control antibody using our recommended optimized protocol. As I said, we don't suggest you try titration on micronucleus amount or time course of digestion time, because we know they are very flexible. And maybe it's a wrong direction to improve your data.
Well, that's our last question, and we'd like to thank you all for attending our Learning Lab Live session on Cut and Run with Dr. Fang Chen. If for some reason your question was not answered today, we will get back to you via email. And if you have any other questions in the future or any technical questions, whether about Cut and Run or any use of our antibodies, feel free to contact our technical team at Cell Signaling Technology. Thank you for attending our Cut and Run session.
Thank you, everyone. Bye. Have a good day.
Bye.